Hello, good evening, everyone. First of all, thank you all very much for being here. Thank you, Caleb, for your willingness to speak with us regarding this important subject. I want to introduce Dr. Caleb. He grew up in the middle of the United States, in Kansas. Sorry. He received his bachelor degree in biology and microbiology from Kansas State University. And he went to earn a PhD degree in genetics from Michigan State University. During his time in Michigan State, he studied molecular mechanism of plant-microbe interaction. After graduating from MSU, he took a position at the U.S. Department of Agriculture in the Agriculture Research Center in Salinas, California, as a research geneticist studying water use efficiency of lettuce and spinach. He joined RiceTech in 2014 as a trades, discovery, and development lead, managing a team focusing on developing new herbicide-tolerant traits of rice, including both full-page and max-ace rice cropping solution. Caleb, thank you very much for accepting our invitation. We are looking forward for your presentation, and it's going to be very nice to hear you.
All right, great. Thanks for the invitation. You know, I appreciate you reaching out to me. I'll go ahead and share my screen here. Thanks for inviting me this evening. Thanks everybody for joining. I know there's probably a few of you that heard me give a presentation a few weeks ago to the weed science group there at the University in Pelotas. So this is going to be pretty familiar for those of you. I did make a couple of tweaks, and using some feedback we got from that, I changed a couple of things. One, I went ahead and switched my title over. So I think the thing to focus on, and what I'm going to talk about more tonight, is kind of the idea of getting started in gene editing for weed management. A lot of the questions that I got last time and the conversations I've had with people have been around the area of, you know, I'm interested in gene editing. I don't know a lot about it from the application side, more maybe the science side. And then kind of, you know, how do I start? How do I apply this to my weed science program? So real quick on my background, which we already covered a little bit. You know, I grew up in Kansas. My bachelor's degree is from Kansas State, biology and microbiology, PhD in genetics at Michigan State. I do want to mention one thing. I was completely a molecular biologist for my entire academic career. So for over a decade, it was all molecular biology. I was a full-time lab person, and I made the transition from lab to field when I went to the USDA ARS facility in California. And that was kind of a joint project where we did some molecular work, but there was a lot of field testing. And that's sort of how I found out that I like doing field work. I like doing more applied work. And then that experience led me directly to industry, to here at RiceTech, where I've been since 2014. I will say transitioning from a vegetable crop to a grain crop is not the normal transition that most people go through. When I came in, there was a lot of questions about why are we hiring lettuce people to work in rice? So that does come up sometime. Don't let them deter you. If that's what you want to do, prove that you can handle it. So I'm mostly going to focus on a little bit of background about what is gene editing. I'll give a little bit of history about the systems. One of the important questions is how is it different from a GMO? And then I'm going to talk about the process. You know, what does it look like if you're going to actually start gene editing in your lab? And then one thing that's often overlooked is how do you confirm if you've been successful? What is a successful edit? And then what do you do with it? I'll talk a little bit about how we at RiceTech are harnessing gene editing technology. And then I'm going to go into the section on weed management with some real world applications. You know, what are people in this field doing right now with this technology? I'm going to share a little bit on kind of how you can find targets. That's a part that's critical and can be a little intimidating, but I have some advice. I'm not going to give lists of targets, but I am going to give you some good resources, I think. A little bit about setting expectations. We're going to go through kind of where this technology is likely headed. And then my favorite part, we're going to talk about licensing and regulation, because that's really the critical component to a gene editing program if you want to go from the lab to a product. Okay, so this slide I've been using for a few years when I talk about gene editing, and I think it's important for us to have a shared definition of, what is gene editing. If you look at a lot of different papers, if you Google it, you're going to get slightly different definitions. You'll get some that are kind of the textbook science, some that are a little bit more towards the regulation. But these three, in my mind, give us the kind of shared idea that we can then use to talk about the rest of this webinar. Simply, it's the generation of precise changes in a targeted DNA sequence within an organism. Or if you want to get more technical, it's the use of a protein and an oligonucleotide to induce site-specific changes in DNA, or the really generic, changing the genes without the introduction of foreign DNA. And that's going to be key, is that what we're doing is we're targeting for changes the native genome of the organism that we're targeting. We're not generally going to be inserting transgenes. We're not going to be inducing permanent insertions of foreign DNA. We're going to try to make small, precise changes within the organism using its own DNA. Okay, so real quick history on gene editing. There are multiple gene editing systems that have been developed over the years. Commonly now in the news, you hear about CRISPR systems or CRISPR-Cas9. That's the latest and the most efficient, kind of widely used system. But there's been a long chain of these systems that have been developed over the years, from meganucleases, zinc finger nucleases, the TALEN system, which is actually what Calyx uses for their product development. And so, all of these have pros and cons. All of them have different licensing structures, and they take different requirements in the lab to be able to operate. CRISPR-based systems, whether it's Cas9 or Cas12 or some of the alternative enzymes, are definitely the most widely used in the current research space, as well as commercial development. And that's for a few reasons. Primarily, they're fairly simple, they're easy to work with, they're adaptable, and they can target a wide percentage of the genome. There are alternatives to Cas9 that are starting to gain more popularity. You'll increasingly hear about those in literature. Specifically within the plant community, Cas12a, which used to be called CPF1, has really kind of taken off. You see a lot of academic research that's focused around that enzyme right now. So just be aware that, all the CRISPRs are similar, but there's subtle differences that may or may not matter for your specific program. And I like to point out that, the developers of the CRISPR-Cas9 system received the Nobel Prize in Chemistry last year for this discovery, which is already making some really big changes, not only in medicine and in genomics research, but also in plant sciences, and it will continue to grow in impact through the next decades. So one important thing that we need to cover is how is gene editing different from GMOs? First, and similar to gene editing, there's not a single definition of what a genetically modified organism is. And so for this definition, I use, a living organism that has been modified through the addition of DNA from a different species. So this is your classic transgenic type GMO. You can also have cisgenics that are GMO, and gene editing regulations change that labeling depending on where you are. But I highlighted 4 major differences that are really the key of why gene editing may be able to be applied to systems where GMOs were not. And those are the mode of modification. With a GMO, you're generally doing an insertion with foreign DNA. And with gene editing, primarily you're making changes internally and you're not permanently inserting any foreign DNA. And that's critical. That turns out to be the critical component when it comes to the regulatory side. Both offer options for trait stacking. That's very common in GMOs, where you'll add insertion cassettes. With gene editing, they refer to it as multiplexing, but you're doing simultaneously editing of multi-loci at the same time. And that's growing in the number that you can target and the precision. What you can target, with a transgene, you can insert anything you want, but you might not be able to precisely control where it goes, things like copy number. With gene editing, the precision within the genome is accurate, but you're limited to changing what's there. Otherwise, you're making an insertion, and then it's the same as traditional GMO technology. The biggest difference is the regulatory landscape. GMO regulations are very well entrenched. They're well established globally, and they vary a lot. Gene editing is still a new technology. It's not mature. The regulatory scene isn't mature. And the regulations vary widely, but generally what we've seen so far is that the regulatory burden from the lab to the field is significantly less. And in several countries, specifically in the Americas, the regulatory burden is very low, just slightly higher than a conventional mutation event. They're not unregulated, but there's a fairly low burden to deregulated status, meeting a few specific criteria, which I'll discuss later. I do like to point out that gene editing is a blanket term. And when we're in the plant space, you see a lot of different abbreviations, and some of these are regional. So I do like to point this out because sometimes, you're talking to a colleague from Europe and they might be using one abbreviation. In North America, we might use something different, and in Asia, it might be different. And so these at the bottom are things that you see a lot, and they generally all mean gene editing. Plant breeding innovation is picking up some steam here in the US. NBTs, or new breeding techniques or new breeding technologies, show up fairly often in Europe, especially in government things coming out of the EU. And then NGTs is another one that we're starting to see more. So it's just good to be familiar with what those are. They're not all different technologies. They're just different names for the same technology. Okay, so a little bit about how the CRISPR system works, and I'm only going to talk about CRISPR because it is the most commonly used system right now. Essentially, you have a sequence of DNA that you are targeting, commonly just referred to as the target. What piece of DNA, what sequence are you trying to edit? Using a guide RNA, which is a short sequence of RNA that matches that DNA sequence you're targeting, it actually localizes the Cas9 protein, which uses a combination of your guide RNA and then a PAM sequence, which stands for protospacer adjacent motif. So it's a short sequence that's within the native genome, and the combination of the distance from the PAM site as well as your guide RNA tells that Cas9 enzyme where to cut. At that point in time, it causes a double-stranded break, and then the cellular repair mechanisms control what your outcome could be. It may be a homologous joining, it may be a random mutation, it may lead to some kind of indel. Your outcome tends to be a little bit unexpected, but the site of the activity is precise. So precise location with a little bit of random activity at the location. The sequence of the PAM site does limit which areas within the genome you're going to be able to target. So you can't target every single base equally well with any of these gene editing systems. Also, each CRISPR-Cas enzyme has slightly different PAM site specificity. And so if you need to edit a specific site, you want to spend the time to look for what kind of PAM sites might be available and whether your enzyme system is compatible. If you're starting from scratch, I would say if you have one target that you want to edit, make sure that the system you're using is going to be compatible with that location. Cas9 is actually a little bit more limited than some of the newer systems, just by the way. Okay, so what does the process look like? My little banner there at the top, you identify the target. That's important. You have to know where you're aiming your editing machinery. You can't just say, I'm doing an insertion, we'll figure out where it goes later. You have to know the precise location that you're targeting. So identify your target. This is critical. You need to sequence the target region in the actual genotype that you're working with. You can't just use a similar genome that you found online. You're working with, in my case, rice. The elite lines that we have, they're not genetically identical in all regions to a lab strain like a Kentucky. So unless you're working with a model organism, you need to sequence the region because guide RNAs are short, PAM sites are very short, and so a single base change can prevent you from being able to target for editing. At that point in time, you design your guide RNA. You're going to want to validate your guide RNA, validate your plasmid carrying the sequence for your Cas enzyme, and then we're actually gene editing. So for rice, and all these images come out of our gene editing lab here at Rice Tech, we generally start with one of two things, either immature embryos or mature seed. But in both cases, we're then using tissue culture to generate callus. We use biolistics. Most labs use biolistics for gene editing, primarily for two reasons. A lot of the protocols were developed using biolistics, and it significantly simplifies downstream regulation in most countries because agrobacterium-mediated transformation works. But since agrobacterium is a known plant pathogen, even though the lab strains are not, it raises red flags and potentially increases the regulatory burden in some countries. And so biolistics generally gives you a little bit easier path if you're trying to develop something for release. At that point in time, we use selectable markers to track successful transformation. Here you see some cells that are expressing GFP. You can see the little green dots. And then you start screening. You can screen with selectable markers like a hygromycin tag, like a GFP insertion, and then back into tissue culture. You're regenerating plantlets from your callus, and eventually you end up with plants and seeds, and that all feeds directly into your screening pipeline. So the actual editing is one little step of the whole process from I want to make a change to I have a plant that has the change. There's a lot of steps and it's a lot of tissue culture. The one advantage of that is if you're a group that already has a lot of expertise working in plant tissue culture, it's pretty easy to adapt what you've already done and add the additional gene editing steps. So you don't have to start from scratch necessarily. Okay, so I can't stress this enough. Think from the very beginning, what is a successful edit? What are you looking for? Sometimes when we think about gene editing, because we know it's precise, we think a little too narrowly. Okay, I'm targeting this location and I want a 2 base deletion. Great. You're probably going to get 40 different changes at the exact site. And so be prepared to not only identify those, but then select the ones you want. You are most likely going to get a small indel of some sort, or you may get a base change. There are systems that specifically do base swaps. Those are often called base editing enzymes. There are Cas systems that you can get that have been modified, or you can build your own. There's also options to use like a homology directed repair, where you're actually giving a template so that the repair is more likely to follow a sequence change that you're desiring. So the overall editing efficiency matters by which enzyme you have. It matters which crop system you're working in. So there's a lot of changes that you're looking at on the overall efficiency. And here I pulled this figure out of a paper just as a nice illustration of what you may see. In this case, the author was targeting this exact sequence site that you can see the ends on both ends of this graphic, but each one of these was a unique editing event that happened with the same guide RNA and the same protein. And you can see there's some very small deletions, there's some much larger deletions, there's some random insertions, there's a few bases that have been swapped. And so, expect a variety of edits and make sure that you have the capacity to figure out which one you want. You need to be able to screen the genotype and the phenotype after you do an edit to make sure that it's what you need. Okay, so how can you detect if you have a good edit? I just mentioned that it was critical. There's a lot of different methods, and some of them depend on what kind of edits you're doing. Some of them are tied to the overall efficiency. Some of them are tied to things like, do you need a specific base change, or do you just need to disrupt the gene? If you only need to do a disruption, an indel or an insertion or a deletion may be exactly what you need, unless you get bad luck. It's a three nucleotide deletion. You just shift your amino acids and it's still a functional protein. And so from kind of the simplest to the most complicated and the most sensitive, things like digestions, a T7 endonuclease digestion or restriction enzyme digestion are pretty basic. A restriction enzyme only works if you change a restriction site in the edited location. And so that one's kind of just luck whether you can even use it or not. But you're already sequencing the target, so you know whether you have any restriction sites there. Sequencing is very useful, either just traditional Sanger sequencing or use of different NGS systems. Sanger sequencing has a fairly low sensitivity, and so you need to make sure that your editing enzyme has a really high efficiency for Sanger to make sense. Now, there are a couple of methods that are really starting to show up more and more in labs that are doing a lot of gene editing, and those are ORNI PCR, which essentially uses a short oligonucleotide to block the site, and then you lose that blocking if you make a change. That's got fairly good sensitivity. It's relatively inexpensive. You can make it high throughput on certain machines. And really the most sensitive for gene editing, which is best if you're doing things like single base changes or SNP editing, is digital drop PCR, ddPCR. Now, if you're at a university that has one of these systems, or if you work for a group that has one of these, fantastic. It works great for gene editing. If you're a university lab that maybe is on your own and needs to purchase one, just a warning, they're expensive. They can do a lot of things. They have a lot of power, but if you're only going to use it for gene editing, it's a significant investment up front for one piece of equipment. Also, the method you use for analysis can impact your accuracy. These are just a few examples of some different software methods we've used to do analysis. And sometimes you get slightly different outputs, even though your sequence data may be the same. The algorithms that interpret that data for editing may give you various degrees of certainty. And so just be aware of that. If one of them doesn't work, maybe a different software package gives you a little bit more accurate results. And then finally, you're going to need to phenotype anything you have. I'm a geneticist, so I'll claim this. Sometimes people that like genetics say, we don't even need to look at the plants. The sequence is everything we need to know. Now, I've been doing applied work long enough now. I want to see the genome, and then I want to see the phenotype to prove that the targeted phenotype we expected is the targeted phenotype that we received. And so in the case of RiceTech, we have containment greenhouses that are closed system greenhouses. And so we do our phenotyping in a closed greenhouse environment. Okay, so just a little bit about RiceTech. So what are we doing? So when RiceTech decided to onboard gene editing technology, it was about 4 years ago, and I was approached by management, given my background in genetics and my current role doing trait development, to start looking into, hey, we've been hearing a lot about gene editing. Is this something that RiceTech should be spending time and energy working on? And so that started over a year-long process of us just kind of getting acquainted with what's available, what systems, what vendors, what licenses, what regulations, and what can it do? What can we do with it? What do rice consumers want? What do rice mills want? And at the end of that process, we decided to move forward with onboarding the technology internally. And so we set up a lab, we started hiring staff, and we signed a license. In the case of Rice Tech, we licensed a system from Benson Hill, which is a company based in St. Louis, Missouri. It is a CRISPR-like system called CMS1. And we are working under a research-only license. And so I do want to emphasize that we are not currently developing products with gene editing technology. What we are doing is we are building a complete gene editing system by adapting this CMS1 technology to our elite germplasm. And that has been a lot of work. And that's some of the warnings I want to share. There was a tremendous amount of work in CRISPR systems that was done in rice, but it was done in things like Kentucky, which are well-characterized lab strains. Those are very, very different than commercial elite germplasm. And so, don't expect that you'll buy the box and you'll use it the first time on your crop and it's going to work. You're going to have to adapt. They're basically giving you all the parts to build the tool, but then you have to build it to fit your system, regardless of which system you get into. Now, if you are working in a model organism, let's say your lab's doing research in Arabidopsis, you can probably buy a system and find protocols online that will work with an Arabidopsis system. But if you're working in any crop, if you're working in something that is not well characterized, you're going to have to do some development work. So just be prepared. And even if you're not doing commercial development, gene editing can be a really powerful tool for testing trait concepts. Because you're not relying on random mutagenesis, you can make targeted changes and then see what the phenotype is. Okay, so pivoting a little bit to kind of the core of I think what this audience is interested in. What can we use gene editing for in weed management specifically? The obvious one, and the big flashing light, is herbicide tolerance traits. Herbicide tolerance is great. It's been a powerful tool for growers to use worldwide. It's solved a lot of problems. Also, we've seen herbicide tolerant weeds show up, so we have new problems to address. One good thing about gene editing for herbicide tolerance is the fact that a large number of tolerant weeds have developed over the last decades gives us a gene pool that we can start mining for new sources of tolerance that may already be characterized. With gene editing, we don't have the concern that we have to be able to cross that into our target species. And so just knowing which genetic changes led to the tolerant weed could potentially give us a tool to apply on the crop side. Gene editing also allows for generation of novel herbicide tolerance. We don't have to rely just on high frequency random mutations or, we get really lucky. You can apply things like machine learning algorithms and computer modeling systems to identify potential novel SNPs for HT targets. We're really coming together in technologies here, which as big data analysis gets better and gene editing technology gets more precise, we're going to increasingly see computer systems that can predict targets that then we can go in the lab, create, and probably within about 1 year start phenotyping in a greenhouse. There are already groups that are doing this. There's several academic labs, and there's a couple of commercial groups that their business model is specifically generating novel mutation. And so we're going to see new sources come up at an increasing rate. One real good benefit is you don't have to physically acquire the source of germplasm. You just need to know the sequence. This can be great if it's a situation where it's not going to be a compatible species, or if you have some kind of import restriction. You're living in a country where you can't import a specific species. In the past meant you had no ability to acquire that genome. With gene editing, you just need the sequence. That's really helpful. In the United States, importing rice seed is challenging. It has to go through a one cycle quarantine process. And so, even for us that operates globally, it makes things a little bit easier if we just need the sequence and not the physical seed. It simplifies things. Also, there's no need for sexual compatibility if you're just trying to use the sequence. which is really helpful. It's not just a situation where, well, we need, a bridge species so we can do a bridge cross or 2 bridge crosses. No, not a problem anymore. We can insert the changes we need directly into our target species. And one of the most powerful things, and if anybody listening has done any trait integration by backcrossing, linkage drag can be a significant problem. Every once in a while you get one of these traits that looks good, and it happens to be right next to something that's really deleterious in the trait donor. And you may not be able to break that linkage if it's too close. With gene editing, you can introgress traits from the rest of the gene germplasm, but you have no linkage drag. If you're looking to edit a single SNP, your final product is 99.99999% exactly the same as what you started with, except for that one SNP. So that's a huge benefit, especially if you want to try to move multiple traits, because every time you move something, if they're on different chromosomes, you have different linkage problems, and you can end up with a messy genome pretty quick. Okay, so just highlighting some real world examples. These are headlines I grabbed from different research papers showing kind of what's going on out there. Obviously, herbicide resistance is a hot area for genome editing. Here's several that are being done. There's one, IMI tolerance in Arabidopsis, adding HT to sugarcane. One that I really liked is, here's an example of HT watermelon. One thing with gene editing is, because the cost to develop is so much less than like a transgenic, it lets companies and universities and institutes develop things that may not have been financially feasible in the past. So you can bring traits to orphan crops. You can bring traits to specialty crops. Also, in some regions, there's not public acceptance for GMO fruits and vegetables, even if there is for grain crops. And so with gene editing and its regulatory status as non-GMO, it has the ability to bring some of these systems, which provides more tools for growers that they wouldn't have had in the past. I know from my time working in lettuce and spinach, consumers don't want GMO lettuce. But as far as we know, gene edited lettuce might be acceptable. I don't know what their current market analysis looks like in lettuce. I've been out of that business a little too long, but GMO lettuce was a complete no-go. Okay, so this is a good chance for us to ask this question. You've been listening to me now for about 30 minutes. And maybe you already thought this, maybe I convinced you, but how do I get started? If you're in academic research, that's a fairly simple path, and I can offer the most advice in the academic side. A lot of the CRISPR-Cas9 systems and other CRISPR systems are available with a research license that is completely free for academic research purposes. And so your barrier to entry, if you're in academia, is essentially free if you already have basic molecular biology equipment. You don't need a lot of specialty equipment unless you need to get a gene gun if you're doing biolistics. But if you're doing academic research, you already have a lab, and you're not concerned with potential product regulation, use agrobacterium-based transformation. You'll save yourself a lot of money on a gene gun. You probably already know how to do it. And it doesn't significantly change the efficiencies you're going to see. And so, a CRISPR system with a research license, it's cheap. It's an easy way to get started. Collaborations are great. Find a lab that's already doing this and partner with them. Having somebody that can help walk you through the initial startup, it makes it a lot easier. They're not just you buy the box and it works the first time. It's not a Qiagen extraction kit. You don't just follow the instructions and you end up with what you want. There is also starting to be a handful, and this is a growing space, of fee-for-service labs where they can do the editing for you. So if you only care about, maybe a single specific change, there are groups that you can give them a target, you can give them your germplasm, and they return you seed that has your intended change in it. These are expensive, and so they might not be a good option for academic research, but it is something that's worth looking into, just as another option. Now, there's a big difference between research and product development. You know, you need a license, you need rights. If you're only doing academic research, you can buy the box and it comes with a research license. You're good to go. If you are doing product development, whether it's academic or commercial, if you want to sell a product at the end, you need to start thinking about the rights implications up front. Just for an example, in the US, the CRISPR-Cas9 system can be licensed either through Corteva. They have a CRISPR consortium and they can give you the blanket patents, or you can go directly to the patent holder, which in the United States is the Broad Institute, and you can license the systems directly. They offer commercial and research only terms. They're not cheap. And so from an academic standpoint, unless your goal day one is we're going to sell a product, you're better off with a free and open academic research license. It will save you hundreds of thousands of dollars. And now we need to find a target, because target is critical. The good part about gene editing is it's precise. The bad part is it's precise. So you have to know what you're looking for. Identifying targets. Great place to start, especially when we're talking about, you know, weed science, weed control, herbicide tolerance traits, is public databases. Here's just a home screen or a screen capture from the International Herbicide Resistant Weed Database. Like I said, you know, herbicide tolerant weeds, they're always our nemesis as people that are working in production agriculture. But now we can start using some of that genomics information and essentially the random mutations that led to tolerant weeds, we may now be able to harvest for our own benefits, fighting against some of those. Now, obviously, we can't take an ALS tolerant trait from a tolerant, you know, barnyard grass and then put it back in a crop, plant it in the same field and control it. But we may be able to, you know, target a mechanism from a terrestrial crop and apply it to an aquatic crop like rice, or vice versa. Or we might be able to shift new sources of tolerance to regions that don't have those tolerant weed species. And so public databases are a fantastic place to start. They're great resources either for specifics or just getting ideas to see what, you know, what might work. Academic literature is always fantastic, but there's so much literature around gene editing and, you know, things like herbicide tolerance traits. It's a little overwhelming if you just start typing in gene editing, herbicide tolerance. And then one that I like to point out is increasingly proprietary databases and genomic resources are gaining popularity. Within industry, these are getting more and more common. And partially this is because of an increased push for regulation of what's called DSI, or digital sequence information. If you've been following any of that discussion lately, the more there's going to be regulations around sequence, the tighter the control of that sequence is going to be. And that's going to affect everybody from a university all the way, you know, through the biggest multinational ag companies. You know, when resources become more expensive, they're going to be more tightly held. Okay, so just this is the warning slide. Here's a few challenges, and setting your expectations is important. CRISPR-Cas systems are not point and shoot systems. There is a learning curve, even if you're an experienced molecular biologist. You're going to have to continually optimize. You're going to have to spend time adapting to your elite lines. Like I mentioned, collaborations can be really helpful, because if you have somebody, even if it's just an academic colleague or a friend that has done this and you guys can chat on the phone every once in a while, it makes things go a lot smoother if you have some help. Reading the papers are great. Reading the protocols, watching videos, watching lectures are fantastic, but having some firsthand experience is a major help. Same way with the tissue culture component. You're gonna have a lot of challenges with the tissue culture part. And so if you have expertise or have access to tissue culture expertise, that's gonna reduce your burden significantly. And like I mentioned, you're gonna have to adapt this to your system. The farther the plant species that you're working in, the farther away it is from any of the model systems, you're likely going to have more challenge. If you're working in a monocot, there's been a lot of work in CRISPRs and monocots. There's been a lot in dicots, but some of the systems are different. If you're working in a system that's vegetatively propagated, that can create its own challenges for good and bad. One, let's say you're working in bananas. You're probably good at doing tissue culture, but it means you have no option to ever go to a seed generation. And so just be prepared for these kind of challenges. Even in rice, where there's a lot of work, we had to spend a lot of time adapting it to our system. And just know, confirming your edits is going to take a lot of time, energy, and expense. And so the editing is the quick part. The confirmation is going to be slow. And so just be prepared for that and plan ahead. It's very easy to edit 5,000 pieces of callus. If you end up with 1,000 plantlets, you now have to screen 1,000 plantlets. One word of caution around that. When you use biolistics and you do transformation on callus, each cell could potentially have a different edit. And so as you regenerate plantlets, if you're regenerating multiple plantlets from the same piece of callus, you need to screen all of them. They might be different. And we have found with CRISPR-based systems, because they retain the plasmid that carries the CRISPR and the guide RNA sequence through the M0 generation, when you harvest your M1 seeds, they might have a different edit. So make sure you confirm at that stage as well. And so be ready. Your pipeline gets really big really fast. And then licensing and regulations can be a challenge. It's a bigger problem if you're planning on commercial products. And so, I'm going to mention that a little bit on some subsequent slides. Okay, so we're thinking long-term here. HT traits are the obvious thing that jumps to mind when you think, okay, how am I going to apply gene editing to, a weed management strategy? But beyond that, there are things that are really going to enable some new options beyond just, okay, I can make a change here and get ALS tolerance. I can make a change here and maybe I get a glyphosate tolerant line. Those are characterized, those are known. Some of these that I listed here are already in progress, and some of them are a little bit farther out. One of them that I refer to as brute force discovery is rapidly becoming something that's a reality. There is at least one company in the US that this is their business model. What they do is they're harnessing some really big data and machine learning systems to identify either entire gene families or entire metabolic pathways from beginning to end for editing. And so what they do is their software systems tell them, or maybe we know through prior research, the endpoint of this metabolic pathway can give us herbicide tolerance. So then what they do is they target every single base of every single gene throughout the entire metabolic pathway, which could be dozens, if not hundreds of different genes, and it could be millions of specific bases that they're targeting for editing. Then they generate just tens of thousands of independent edits, and then they phenotype everything. And so this is incredibly labor intensive and time intensive, but if you get something out of this, it's got a really good chance of being novel. And that's what we really need. We know when fighting tolerant weed species that are always coming up, we need new tools. The rate of discovery of new herbicides, new mode of actions is really decreasing, and it's slow. And so we need new tools to fight back for the farmers. Complex multiplexing is becoming more and more a reality. There are groups now that are targeting dozens of simultaneous edits in multiple genes at the same time. So one editing event, and you get a large number of changes all over the genome. There are some groups that are working specifically to optimize the number of edits you can target simultaneously. And it seems like fairly quickly we'll be able to edit hundreds, if not thousands, of unique bases at the same time. And then kind of I challenge everybody in this call to think a little bit about, well, given that gene editing can target anything, and we don't just have to select by spraying a mutant population and looking for tolerant plants, what are some other phenotypes that may give us some benefits in a really integrated weed management system? And so some things could be, more vigorous early plant growth. What about if we could alter leaf angle so that the canopy closes earlier, and so we have a earlier season shading between rows? Another one that I think is really going to be critical and is already being worked on in gene editing is if we can improve abiotic stress tolerance, that might give us some really big shifts in available crops for a better rotation schedule. There's a lot of farmers in rice that grow rice every year because their land's really only good for rice. Well, that significantly limits their weed control options from the chemistry standpoint. Well, what if we could develop a soybean that could handle partial season flooding? Or what if we could add herbicide tolerance to some vegetable crops and start rotating these in? And so we really need to think beyond just herbicide tolerance when we're looking at how we as scientists can apply gene editing technology as part of, more of an integrated approach to weed management. Okay, so I've say now that this comes the less fun part. I spend a significant percentage of my time on things that have to do with regulations. And so even though it's maybe a little less fun to think about as a scientist, it's critical if our goal is to develop something that a farmer can use, something that's going to benefit the end consumer. The patent and rights landscape around gene editing is complicated, and that's to put it nicely. It varies globally, it varies regionally. The patent rights to the enzyme systems and the regulations on the products are different. And so just, prepare yourself. And so I'm gonna just briefly touch on what is referred to as freedom to operate, or FTO. For some of you, this is probably something that you rolled your eyes and said, oh no, not more of this. And for other listeners, this may be something you've never even heard of before. But what this is it's the ability of scientists to access and utilize the technology in this case. It's not something to be scared of, but it's something you want to be aware of. As I mentioned, if you're an academic researcher, you can generally freely use CRISPR-Cas9 for research purposes with no concerns with freedom to operate. You essentially have full freedom to operate. Now, there's also other CRISPR systems that you can access. Now, even if you're a academic group, if your goal is to develop a product and you work for a department that specifically wants to release varieties of your crop to local producers, you need to think about that up front, because there may be a different license that's required throughout the development process if the end goal is commercialization. You're not going to be blocked from using it, but it may present challenges downstream. And so it's just good to think about it up front. If you're a commercial operation like us at Rice Tech, and you either think or may someday use it for license, or the fact that you're a for-profit business, you really need to get some kind of commercial structure in place just to cover your bases. It's hard to convince rights holders that you're only doing research, even in our case, when we're only doing research, if you're a commercial business. Patent ownership is also really complicated around CRISPR systems. There are some patents that were issued to different groups in the US versus Europe, for example, and that ownership structure is not completely solved, so may be subject to change, just a warning. Okay, product regulation. So when I say product regulation, I specifically mean, okay, we've completed an edit, we've phenotyped it, and now we want to try to go to market. So it's not the enzyme systems, it's the product of the actual edit. This is completely separate from the licensing of the systems. Regulations are determined at the level of the country you operate in, or the country you sell in, or both. Like I mentioned, it's based on local jurisdiction. The degree of regulation varies wildly. From some countries where there is really no unique regulations, all the way to the European Union and New Zealand that have banned the technology almost completely. And so, your experience may vary when it comes to regulating gene editing products. Generally, foreign DNA is your biggest problem. So you do not want foreign DNA insertions in your final product. Generally considered best practice is to sequence the entire genome of your final edited line at the point that you want to try to commercialize it. Generally, it's recommended to use some kind of NGS methodology with a very high coverage and overlap rate, and then try to assemble it and see what you have. The biggest problem and what has tripped people up in the past is backbone insertion of your plasmid. I put the copy of the cover of Wired magazine here with the cow as an example. A company brought a trait in a dairy cow to the market in Brazil that was going to provide for Holstein cattle that were naturally polled, meaning no horns, which prevents the cows from having to be dehorned when they're young. They did this through editing in a change that was present in some European breeds of beef cattle that were naturally hornless. What happened was this company did not do full genome sequencing. They used sort of the transgenic method where they targeted their known sequence and they looked to see if they could detect it across their genome. They were not able to detect it. They got ready to go to market. And then whole NGS sequencing revealed that a very small fragment of their plasmid had actually randomly inserted in the genome. So what they had was not an unregulated gene edited cow. It was actually a GMO regulated cow, which was not allowed on the market. And they had to pull their technology from the market. They got a lot of bad press. I think what they're doing is really good. I think that they'll get there. They're working on it right now. In fact, they actually have another cattle trait for heat tolerance that was developed, and they double checked that. There's no foreign DNA, and they're still planning on coming to market with that in the future. I put a website up here. This is a good resource for checking kind of the overall regulatory situation. It's part of the genetic literacy project. This map is kind of just a real overview. It came off of this website, I guess it was in January, so it's been a while now. Just kind of showing you the differences globally. Generally, the Americas have regulations in place that are much more friendly to gene editing. Whereas Europe right now, with a European court ruling, has essentially banned GMOs. India, China are still working on their regulations. Australia has favorable regulations, and New Zealand has blocked the technology. So it really depends on where you are, what kind of regulations you're looking at. Okay, so here's a couple of scenarios that I wanted to talk about just to get people thinking about kind of how the regulatory scenario may affect them. I borrowed this little graphic from a SCRA workshop a few months ago, and it was from Carrie McMahon with the FDA, Food and Drug Administration here in the United States. And really what you need to think about if you're talking about how is my final product regulated, and here's a couple of examples. Let's talk about a tomato. In this case, our tomato is going to be grown for fresh market purposes. We're going to distribute the seed to local growers, and they're going to sell the tomatoes at local markets, and they're not going to use the residual plant material for anything. At the end of their crop season, they're going to till it under, and they're going to plant a new crop the next year. And so in this case, you're only worried about how that product may be regulated in your local jurisdiction. And so if I'm bringing this tomato to market in Brazil, I only am concerned with the Brazil regulations. It's not going to be exported. It's not going to be, there's not alternative uses. And so I only need to worry in the case of Brazil, does it have foreign DNA? Let's say this was a small deletion, no foreign DNA, small change. We're not going to have any problem getting this deregulated and onto the market in Brazil, but we're not going to be able to go on the market in New Zealand. Just as a side note, about 3 weeks ago, a gene edited tomato, the first one in the world, has went on the market in Japan. And so this is a reality today that there are gene edited tomatoes available for sale in Japan. Second scenario is something like wheat. In this case, now I'm a multinational. I've done a gene edited wheat, and I want to sell it to every region of the globe that produces wheat. And so now I have to worry about what is the regulatory scenario for planting a gene edited product in all major wheat growing countries. So this is going to be complicated. Also, the product is going to enter a global commodity market. So it may be commingled with other grain. And so now I have to worry about export markets. And so this is pretty quickly becoming more of a problem and more of a challenge. Also, with wheat, there are multiple potential food uses. Maybe we're not just using this wheat to produce flour, but some of it's going to be used for extracting gluten, and the gluten is going to be used for other purposes. And so now we have to worry about what are the alternative purposes and uses, and how are those going to be regulated. And then finally, if there's any use as a byproduct, I mentioned gluten could be extracted. Well, what if it's a situation where we're harvesting or collecting chaff from the field? In the case of wheat, maybe we're baling the straw and it's being sent to a dairy farm for bedding, or maybe it's being mixed with feed. Well, now there's another potential use and another potential regulatory hurdle. And so generally, if it's a local fresh market, a lot easier to deal with. If it's a multinational, complicated. We're in rice. It's definitely a multinational. It's definitely a commodity. Even here in the United States, where we have fairly low regulatory burden for gene editing, we still have to meet a couple of key provisions to get an exemption. And those are no foreign DNA. And that includes indels. We can't have any insertions to fall under the lowest level of regulatory burden. Small deletions are fine. The changes have to be present within the natural genome. In the case of rice, it has to be a rice compatible species. And so, there's just a few things we have to meet to be okay. Okay. So, and I'm running right about on time here. So I just want to, in conclusion, gene editing is going to be the next big technology shift in plant breeding. It's not a fad. It's not going to disappear. It's being incorporated into all the major programs right now globally. It's big at universities. It has really democratized the access to do genetic changes in crops. And I think it's really going to be a great boost for things like orphan crops, things like smaller market crops, things like regionally adapted varieties, because universities that may not have been able to go through the time and expense to develop transgenic products, with gene editing, it's a lot simpler, it's a lot cheaper. It's being widely used. The regulatory environment is changing, but it's becoming more clear. Barrier to entry is low. Specifically with weed management, it's going to bring HT traits to things that 10 years ago we never imagined was going to be an option. We're going to see that fairly soon. We're going to finally be able to generate a lot of novel herbicide tolerance mechanisms, which we desperately need. And it's going to increase the rates of discovery for sure. So I see a lot of potential here, and hopefully it works this way. But there's some really promising early signs on it. And with that, I will stop for questions. I work for Rice Tech, so here's our website. For those of you that don't know about Rice Tech, we're a hybrid rice seed company. We're based in Alvin, Texas, outside of Houston. We are global. We operate out of 3 regions, primarily in the United States, in Brazil, and in India. And so these are screenshots from our home pages here, just as an example. So with that, thanks for your time, and I'm happy to take questions if we have some.
All right. Thank you, Caleb. It was an excellent talk. We have 4 questions, and I will read it to you. Julian Oliva, is there any experience on gene editing in order to improve P450 or glutathione in order to improve selectivity of herbicides, post-emergence herbicide, example in sorghum?
Okay. I can't point to a paper specifically that's around like P450 metabolic mechanisms. But just like anything with gene editing, there's a lot of research that shows that if you can modulate expression of P450s, you can get kind of a general enhancement to tolerance to a variety of classes of herbicide. Now, we may not understand every point that we can manipulate to get those outcomes. And so therein lies the challenge. And those type of mechanisms are perfect for some of these kind of bioinformatics methodologies, because something like a target site mechanism where we need to make a steric change to the protein, fairly easy to accomplish with a single change. When we're trying to manipulate overall metabolism, sometimes it's more challenging than either just a simple deletion or disrupting of the gene. We may need to be able to increase expression or reduce expression, but it could also be a situation where if you knock out expression, it's potentially lethal, or it comes with significant fitness costs. But I would encourage you, if you're interested in that, I think there's a lot of potential there. The idea of combining target and non-target site mechanisms seem to have a lot of promise. And, I think we would all love to figure out, exactly how we can apply a quote unquote internal safener, so to speak, by improving overall metabolism across not only monocots, but if we could bring that kind of technology over to dicots, that would be fantastic.
Thank you. We have another question from Cyrano. Gene editing will be more efficient than transgenic technology for the point of view of herbicide tolerance? That's a question. Does the regulation can help on that, or is this still early to say?
I probably wouldn't use the word, is it more efficient? Here's the biggest differences when it comes to actually bringing products to market. The regulatory burden is significantly reduced. I think we've all heard the kind of numbers about the years and dollars it takes to bring a transgenic trait to market. And that has excluded anybody that's not a large multinational from being able to bring something to market. With gene editing, you can likely bring a product to market for, this is a complete guess because we have not brought a product to market and we're not currently working on commercial development. But instead of 40 million for a transgenic, it might be under 1 million to bring it to market for something like gene editing. You're a little bit more limited because we don't have the option like glyphosate tolerance transgenics to bring in an entire gene or a gene variant, but things that we already know. We have a lot of SNPs that we know we can mutate within an ACCase protein and get an ACCase tolerant version of that protein in an ACCase tolerant crop. And so it lets us come to market faster. It lets more groups bring products to market. One thing that's really good with gene editing that we couldn't do with transgenics is it allows for something that has a lower return on investment in the market still be a viable product. And that's where we're going to see a huge advantage, because some of these crops that couldn't support 40 or 50 or $100 million development costs, they can support, $700,000 development costs. And so you're going to be able to see the technology applied more broadly. It tends to be a little bit faster to market because of primarily because of the regulatory time. And so, I'm not saying it's more efficient in the genetic component, but I think it's going to be more broadly applied, and it has a higher probability of coming to market eventually.
Mm-hmm. That's interesting. Cyrano, another question: what could be the most interesting herbicide chemistry group to use as a target to develop genetic tolerance in rice through gene editing? What would be the chemical group that you would target?
Jeez. I'm at a huge disadvantage here because I'm probably the only person on the call that has no background in weed science. I had never worked with herbicides a day in my life until I came to Rice Tech. I'm going to give a really evasive answer. I think the real answer is not there's a perfect chemistry group, because there's always a challenge with having enough systems that you can allow a farmer to be able to rotate to maximize their integrated weed management strategies. And so if I was saying what the best option would be for, I'm in rice, but for any crop, is really if we could have one tolerant product to every class of herbicide, farmers would be able to rotate and solve any one of their problems with chemistry. Now, with rice being an aquatic, there are some classifications that are essentially a no-go due to things like aquatic organism toxicity. But, you know, I really don't have like a specific answer of, okay, this is the one that's going to solve all the problems and we're done. I know if you look at the literature that's available, the ones that are most well characterized are what are getting the most effort right now. There's a lot of papers where people are working on gene editing ALS tolerance. There's a lot of papers on ACCA class tolerance. There's a lot of papers on glyphosate tolerance. There is one trait, and I actually don't know who submitted this, but in the United States, there is one trait in rice that has been submitted for addition to the gene edited exemption list. That's for glufosinate tolerance. And so, you know, really any herbicide mechanism that there's any SNPs that are known, those are all being worked on right now, really across the academic spectrum. If you guys want to see what the whole effort of gene editing and herbicide tolerance looks like, go on Google and type in gene edited herbicide tolerance, and you're going to get tens of thousands of hits. And it's just really a tremendous number of groups that are working on this. In my crop rice, you're seeing dozens of universities in China that are putting a lot of effort in developing traits because they need to, you know, produce more grain. They have a growing population. We're all dealing with those kind of challenges. Yeah, I know within the academic groups in the United States, all the universities that have rice breeding programs are working on gene editing, and they're specifically working on gene editing of basically two things: trying to increase yield and trying to bring herbicide tolerance traits into their lines.
Yeah. All right. We don't have any other question. With that, I thank you very much, Caleb, for your presentation. It was very interesting. We have several good insights on the and very you share with us your hands-on experience on that, and that's very good. And sometimes you read the paper and you don't realize how much you need to go through to finish up or to really do the gene editing, and you brought several interesting insights on that. We appreciate that. And this is our last webinar in our webinar series, and I really thank you very much for accepting our invitation and to share your experience. And with that, we say goodbye to everyone. Thank you, Caleb. Thank you all very much for being here. And we hope to see you next year or the year after that, then when we have another webinar series. Thank you. Thank you, Caleb.
All right. Thanks for the invitation. I appreciate it. And if anybody listening has the answer of what the perfect rice HT system is, please send it to my email. I would love to know it.
That's true.
All right. Thank you.
Thank you. Bye-bye.